The innate immunity is our first line of defense. When a pathogen enters our body, innate immunity try to prevent the pathogenesis or try to kill the pathogen. The first component is anatomical barrier. This anatomical barrier can be physical and chemical. There are cells of innate immunity: macrophage, neutrophil, NK cell. So these cells, basophils, these cells actually perform two important functions. One is phagocytosis and production of cytokine and chemokine. The cytokine that produced by these cells are actually IL-1, IL-6, IL-18, IL-12 and TNF alpha mainly. Chemokines are some molecules that attract immune cells at the site of infection. There are some important receptors called pattern recognition receptors. Can recognize PAMP, a molecule, a signature molecule of pathogen. Full form of PAMP is pathogen-associated molecular pattern. Examples of pattern recognition receptors are toll-like receptors, C-type lectin receptor, retinoic acid-like receptor and NOD-like receptor. These three receptors. The other components are inflammasome. This is a supramolecular complex and complement pathway. The first is anatomical barrier. Our body actually have this anatomical barrier. Like you can see, skin, gut, lungs, eyes, nose and oral cavity. These are all anatomical barrier. So they provide anatomical barrier by two ways. One is physical or mechanical and another is chemical. Physical means you can see the epithelial cells joined by tight junctions. So epithelial cells are very tight, associated with tightly, so that pathogen cannot enter. But if there is any injury, this tight junction can be break down. Longitudinal flow of air and fluid and movement of mucus by cilia, tears, nasal cilia. These are all mechanical barrier. There are also chemical barrier like skin contain fatty acids in the sebum and also beta defensins, lamellar bodies and cathelicidins. In my phagocytosis class, I have mentioned that two important antimicrobial peptides you must remember. One is defensin and another is cathelicidin, because these two molecules are also expressed by phagocyte like macrophage, dendritic cell and neutrophil. Epithelial cells also produce beta defensins, lamellar bodies and cathelicidins. In the gut, there is low pH, mean acid in nature. Enzymes like pepsin are there and chemicals like alpha defensins, cathelicidins. Chemical barrier in the lung is pulmonary surfactant proteins. Surfactant proteins like SPA and SPD. These are also acts as a opsonin and also contain alpha defensin and cathelicidin. In the eyes, there are chemicals like lysozyme and enzymes and histamine and beta defensins. E. coli bacteria cannot grow in our skin. Scientists have found one chemical called psoriasin, and they have also observed psoriasin is very specific. It can prevent E. coli but not the Streptococcus aureus, another bacteria. So you can see two individuals actually inoculated with E. coli and S. aureus, and after they put their fingers in the culture plate, what has been seen? E. coli do not grow but S. aureus grow. So psoriasin actually prevent E. coli, not S. aureus. So psoriasin chemical is very species specific. Next term is PAM. PAM means pathogen-associated molecular pattern. The signature molecule of pathogen. It can be nucleic acid like ssRNA of virus, double-strand RNA of virus, CpG of virus bacteria. It can be protein, pillin, flagellin, cell wall lipids like LPS, lipopolysaccharide in gram-negative bacteria and lipoteichoic acid in gram-positive bacteria. Gram-positive bacteria do not express LPS. Carbohydrates like mannan and glucans, these are all PAMP. Then there are some receptors that can recognize this PAMP. These are called pattern recognition receptors like toll-like receptor, NOD-like receptor, retinoic acid, inducible gene 1-like receptor. You can simply can remember as a retinoic acid receptor. CDS means cytosolic DNA sensors that can sense bacterial or viral DNA. C-type lectin receptors. These are the receptors. Scavenger receptor and another important receptor called N-formyl methionine leucine phenylalanine receptors. So this receptor can recognize this peptide N-formyl methionine leucine phenylalanine. Not only that, this receptor can only recognize N-formyl methionine because this molecules or amino acid is only present on bacteria, not by any eukaryotes. So these are all pattern recognition receptors, and there are some soluble molecules like pentraxin family molecules called C-reactive proteins, lectins, and in this group manage binding lectin surfactant proteins like SPA, SPD are components, example phycolin and complement pathway. Innate and adaptive immunity, the two interconnected arms. Two interconnected arms are bridges by dendritic cells. You can see here adaptive immunity cells like B cell, T cell, and innate immunity like neutrophil, eosinophil, basophil, and monocyte and macrophage. But dendritic cells actually bridge these two immunity, innate and adaptive. Another component of our innate immunity is cytosolic DNA sensor called CDS. So when virus or bacteria enter into the cell, a molecule called C-gas, CGAS means cyclic GAMP synthase. This molecule remains inactive, but when virus or bacteria enter, it becomes active, and it can sense the double-strand DNA of virus. And in our body, ATP and GTP are present. They actually become cyclic ATP, AMP, and cyclic GMP, and these two molecules bind with each other to form cyclic guanosine adenosine monophosphate. This is produced by virus DNA. Bacteria can also produce cyclic di-guanosine monophosphate and cyclic di-AMP. Bacteria do not produce CGAMP. So double-stranded DNA from viruses activates C-gas to produce CGMP from ATP and GTP. The next is CGMP or other bacterial derived cyclic dinucleotides. This is CGMP, and these are the dinucleotides. Bind to STING, another receptor present on the endoplasmic reticulum called STING. It means stimulator of IFN gene. So this receptor actually induce IFN alpha and beta, that are type 1 IFN gene. So when CGMP or other bacterial derived cyclic dinucleotides bind to the STING dimer present on the endoplasmic membrane and activates its signaling. So STING activates the kinase called TBK1 to phosphorylate IRF3, a transcription factor. Then it can enter into the nucleus and helps to transcribe type 1 interferon gene. Type 1 interferon is IFN alpha and beta. These two molecules, when produced by a cell, develop antiviral immunity. As I have mentioned in the first slide, cytokines can be secreted by cells. Example of the cytokines are pro-inflammatory cytokines like IL-1, IL-6, IL-12, IL-18, TNF alpha. Here I am only showing a function of particular interleukin called IL-6. So bacteria induce macrophage to produce IL-6, which acts on hepatocytes, liver cells, to induce synthesis of acute-phase proteins. Why it is called acute-phase proteins? Because when IL-6 or this type of cytokine are secreted, this liver very fast synthesize these proteins. The protein concentration increases very high in a short period, so it is called acute-phase proteins. So liver can secrete now serum amyloid protein, C-reactive protein, fibrinogen, manage binding lectin, and SPA, SPD. These are the surfactant proteins. Many of them are opsonate.